So today I'll be discussing on nucleic acid chemistry. Nucleic acids are polymer of specific sequences of monomeric unit called the nucleotides, which are linked together through the 3' 5' phosphodiester bond. So there are two types of nucleic acids, well known, DNAs and RNAs. Nucleic acid serve as repositories and transmitters of genetic information, means all the information is coded in the form of genes over the DNA. Genes are specific sequences, small regions of specific sequences of DNA, which on being expressed through transcription, they lead to the formation of RNAs, in which the information is coded, and further RNA go to the cytoplasm, and through translation they synthesize a specific kind of protein, and the action of protein is then manifested. So this is how they are transmitters of genetic information. And this whole process is known as central dogma of life. Life sustainability based on the type of protein at every step of uh, every vital act of the life. So these proteins can be synthesized through RNA, and RNA which in turn being expressed through DNA. Now, the basic structure of nucleic acid. Nucleic acid consists of nucleotide, which in turn consists of phosphate, sugar, and base. And nucleotide, if we remove this phosphate group, the sugar and base entity is known as nucleoside. So we should keep remember this terminology, very well-known terminology. So nucleoside to nucleotide through addition of phosphate bond, and this then further polymerization of this nucleotide will lead to the nucleic acid. And these nucleic acid, you can see the structure. There is a phosphate sugar backbone is there, and these are the bonds. These bonds in between the phosphate and sugar are known as phosphodiester bond. So through these bonds there is a formation of this phosphate sugar chain, and these are quite hydrophilic in nature. So they are in the double helical structure of the DNA. They are externally or outwardly directed. They are being more hydrophilic. While the hydrophobic bases, they lie interiorly like bases. Bases are in the center of helix. So these bases attach with the sugar. So what are the functions of nucleic acid? The DNA is a chemical basis of heredity. It is a reserve bank of genetic information responsible for maintaining the identity of different species of organisms over millions of years. Cellular function is under the control of DNA, and it is the basic information pathway. DNA directs the synthesis of RNA, which in turn directs the protein synthesis. And proteins are at every vital act of life. So here it is the more clear account of the basic structure of nucleic acid. Polymers of nucleotide held together by 5' 3' sugar phosphate bridge. So this is the 5' end, 5' to 3' end. So this is how the chain grows and polymerization happens. Now, what are the nucleotide? How do they consist of? Already told you they consist of pentose sugar, phosphate, and nitrogenous base. The same thing is here, composition of nucleic acid. So once we nucleic acid is basically the polymer of nucleotide, which consists of nucleoside and phosphate, and nucleosides consist of bases and sugar. So this is the structure of pentose sugar in RNA and DNA. This is the ribose sugar. So what is the basic difference between the ribose and deoxyribose? The main sugar is the ribose, which been deoxidized. So this second 2' hydroxy group is removed, and this it's converted up into the deoxyribose sugar. This is the basic difference. No oxygen bounded to this carbon. And it has got the 5' end, which contains CH2OH group. This free end is there, and this OH group 5' first prime end, at the first prime end there is an hydroxyl group through which there will be attachment of bases. So the backbone of the or polymerization between the sugar and phosphate happens to 5' and 3' hydroxyl group. We will see in further slides. So the two principal purine bases present in DNA are adenine and guanine. So this is the basic structure. This is the purine ring, and there is an amino group present at the numbering is done in an anti-clockwise manner. So we start numbering 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is how we do number in an anti-clockwise manner. So this is the adenine and this is the guanine. So in the guanine there is a ketonic group at the 7th position, and there is a amino group at the 5th position. Here the amino group is at 7th position. Few biologically important bases are uric acid. We all know uric acid, the acid is the end product of urine metabolism. Then hypoxanthine, clinically very important, intermediate of uric acid synthesis. Then xanthine, both are intermediate of uric acid synthesis. So we will see this, the same purine structure is there. So uric acid is another purine base. It is the end product of purine nucleotide metabolism. Other purine bases are hypoxanthine and xanthine. They are intermediates in the formation of adenine and guanine nucleotide. Now, what are the pyrimidine bases? Pyrimidine bases are cytosine, uracil, and thymine. So this is the structure of cytosine. There is a two ring pyrimidine ring is there. The basic pyrimidine ring contains the two nitrogen at the at 1 and 3rd position. So everywhere you will find this pyrimidine ring, but the substituents are different. In cytosine, there is an amino group, and then there is a ketonic group. So this is thymine, structure of thymine. Then this is the structure of uracil. And cytosine is present both in DNA and RNA, while thymine is present only in DNA, while uracil present in the only in RNA. So this is how do we name the nucleosides and nucleotides? So nucleoside like adenine and ribose sugar, so it is adenosine. Then similarly, guanosine. Then uracil and ribose, it is uridine. And then cytosine and ribose, it is cytidine. Then similarly, in the deoxyribose nucleosides, adenine plus deoxyribose sugar, they form the deoxyadenosine. Then similarly, guanosine, deoxycytidine, deoxythymidine. So initial small d represent the deoxy. Now, purine bases present in the plants. Few purine bases are they are biologically important, and they are present abundantly in the plant. So, plant contains certain methylated purine like caffeine in coffee. It acts as a stimulant. Then theophylline is present in the tea leaves. It acts as a bronchial, smooth muscle relaxant. Then theobromine is present in the coca, and it acts as a vasodilator. It lowers the blood pressure. So adenine has an amino group onto the C6 position of the ring. Guanine has an amino group at the C2 position, and the carbonyl group at the C6 position. So purine, this is the structure of purine ring, already explained to you all. So purine analogs present purine analogs of biological and clinical importance. They are number one. They have structural similarities, but inhibit the enzymes involved in the purine nucleotides. For example, allopurinol, it inhibits the xanthine oxidase and used in the treatment of hyperuricemia. Then second is 6-mercaptopurine. It inhibits the purine nucleotide synthesis and used as an anti-cancer drug. So then metabolic intermediates, so these are synthetically produced, and metabolic intermediates are these are formed during the metabolism of nucleotides. For example, hypoxanthine, xanthine, and uric acid. Then thymine contain methyl group at C5 position with the carbonyl group at C4 and C2 position. So naming is very easy. Right from any of the nitrogen we can name 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So how we can remember in uracil there are two ketonic group at the 2 and 4 position. Similarly, in the thiamine there is a two ketonic group at the 2 and 4 position, and in a fifth position there is a methyl group, ketonic or carbonyl group we can say that. Then structure of DNA. DNA is a polymer of deoxyribonucleotides. The monomeric units held together by 3 prime, 5 prime phosphor diester bond as in backbone. DNA is a very flexible molecule and has the ability to exist in various forms based on the environmental condition, a feature known as a structural polymorphism. Then conformation of DNA double helix. The DNA double helical structure exist in 6 forms. Form A, B, C, D, E, and Z form. Among these, the B, A, and Z forms are very, very important. The B form is the most predominant form under the physiological condition. A form is right-handed helix. It contains 11 base pair in one turn. Then A form DNA is tilted from the central axis. So these are the structure of various kinds of DNAs, bDNA, aDNA, zDNA. So double helical structure of DNA exists in these forms. This is the double helical structure of the DNA, consists of two backbones, consist of sugar and phosphate, attached through each other via the bases, opposing faces. One purine base faces the pyrimidine pyrimidine base of the other strand, and this is how the bases of opposite strands are linked through hydrogen bonds. We will see them in more detail. So adenine always joins with thymine. This is the structural complementarity. Only adenine can bind up with thiamine, and similarly, guanine prefers to bind up with cytosine. So, in case of DNA only, there is a Chargaff's rule of base pairing. According to him, the DNA has the equal numbers of adenine and thiamine residues, because जितने आपके adenine होंगे उतने ही आपके thiamine होंगे, because adenine is joined with thiamine only. And similarly, the guanine and cytosine residues are equal. Why they are equal? Because they are only facing each other and bonded through each other via hydrogen bonding. So this is the Chargaff rule. This is rule is known as the Chargaff rule of molar equivalence of purines and pyrimidines in DNA structure. But this rule cannot be applied for RNA, because RNA is not double helical and there is no base pairing in that. Then denaturation of DNA. The two strands of DNA are held together by hydrogen bonds, and disruption of these hydrogen bond, like by change in pH or increase in temperature, will result in their separation. We can see it here by treatment with heat or change in pH, it will lead to the separation of two strands. And on being, these are used to be generally reversible phenomena. So once these temperature is, the sample is cooled down, there is a renaturation. So the phenomena of loss of helical structure of DNA is known as denaturation. Then, but the phosphodiester bonds are not broken by the denaturation process, and it is measured by absorbance at 260 nanometer. So this is how we study the phenomena of denaturation. And what is the melting temperature? Melting temperature is that temperature at which half of the helical structure of DNA is lost. In any of the sample of DNA, half of the helical structure of DNA is being lost. And GC base pair are more stable than the AT base pair. Why? because there are three double bonds between the guanine and cytosine in the structure of DNA, while only two hydrogen bonds are available between the adenine and thiamine. So that's how because of three hydrogen bond, it is more stable as compared to 80 base pair. So the transition melting temperature is greater for DNA with high content of GC pairs. And formamide destabilizes the hydrogen bond of base pair. This is used in recombinant DNA topology, because in DNA topology, we can separate a specific gene and specific fragment. So the treatment of the DNA is being done by formamide. And then renaturation is reannealing, or the separated component complementary DNA are no, they rejoin and form a double helical structure. Now the organization of DNA in cell. So prokaryotic DNA, the DNA is organized as a single chromosome in the form of double-stranded circle and packed in the form of nucleoid. What we call it? This is the circular structure. Circular DNA is known as nucleoid in the prokaryotic DNA. While in the eukaryotic DNA, the DNA is associated with various proteins to form the chromatin, which then organized in compact structures known as. So eukaryotic DNA, in this, the DNA is associated with various proteins to form chromatin, which then organize into the compact structure known as chromosome. Now move on to the types of RNAs. There are three types of RNAs. 5 to 10% messenger RNA, 10 to 20% transfer RNA, and remaining 50 to 80% is ribosomal RNA. So RNAs are synthesized from DNA, and they are basically involved in the protein synthesis. So due to the single stranded nature, the RNA doesn't follow the Chargaff's rule. There is no question of relationship between the purines and pyrimidine contents. Now, alkali hydrolysis, the alkali can hydrolyze RNA to 2' 3' cyclic diester. This is due to the presence of OH group at the 2' position. Then orthogonal color reaction is a specific reaction given by the RNAs, and RNAs can be identified by orthogonal color reactions due to the presence of ribose. Now let's talk on the messenger RNA. It carries the genetic information from DNA for protein synthesis. So its precursor form is a hnRNA, which is also known as heterogeneous nuclear RNA. Now, structurally, it contains two caps at 5' and 3' end. So there is an inverted 7-methyl GTP, guanine triphosphate, attached at the 5' end, while the 5' UTR, that is the 5' untranslated region, is at the 5' end. This 7-methyl GTP is present at the 3' end. So, so there are two caps present at 3' and 5' end. Now, coding region contains three types of codons. One is the initiating codon. Initiating codon is always the AUG, which is meant for methionine. So first, during the protein synthesis, first addition is there of methionine. Then specific codon, they are present for different amino acids. Then terminating codons are very specific, which are UGA, UAA, and UAG. So there is a 3' UTR or 3' untranslated region at the 3' end. Then there is a polyadenylate chain is also available, which consists of 200 to 300 bases. It is also known as poly A tail. So these adenylate residues are present at the 3' end. Messenger RNA accounts for 5 to 10% of total RNA. Then there is a structure of transfer RNA, which consists of 5 arms. So transfer RNA contains 71 to 80 nucleotides. Molecular weight is 25,000. At least 20 species of transfer RNAs corresponding to 20 amino acids are available. and it is required for protein synthesis. It contains many unusual bases and nucleosides. That means there are various derivatives of the basic bases and nucleotides. So unusual bases present in the transfer RNA are thiamine, dihydrouracil, hypoxanthine, 1-methyladenine, 2-nitro-dimethylguanine. Then pseudouridine is an unusual nucleotide found in the tRNA structure. So structurally, it is a clover shaped. It has 5 arms. First arm is the CCA arm, which is present at the 3' end. It is an acceptor arm, which accepts the amino acid and attachment of an amino acid happens through this particular region. Then second is the D arm, which contains the dihydrouracil. Then thiamine, pseudouridine, cytosine arm is present, which contain a pseudouridine as an unusual base nucleoside. Then there is an extra arm. There is a anticodon arm is present, through which there is a attachment of codons of messenger RNA, and this is a extra arm. Anticodon arm contains sequence of 3 bases that are complementary to the codons of messenger RNA. Transfer RNA is also called the adapter transfer RNA because it carries specific amino acids on its 3' end along with the anticodon. So at its anticodon arm. The extra arm, also called as variable arm, and based on the length of extra arm, transfer RNA is classified into class 1 tRNA, which contains a short arm, that is 3 to 5 base pairs only, and there is a class 2T transfer RNA arm, which contains the long arm, 13 to 20 base pairs. So this is about the transfer RNA. Then ribosomal RNA. It is found in ribosome. The eukaryotic ribosomes are factories of protein synthesis. It composed of 2 major nucleoprotein complexes. That is 60 S subunit and 40 S subunit. 60 S subunit contains 20 S ribosomal RNA, 5 S ribosomal RNA, and 8 S ribosomal RNA. 48 subunit contains 18 S RNA. Main function is in the protein synthesis. Then synthetic analogues of nucleotides. Already I have discussed allopyranol, 5-fluorouracil, 6 mercaptopurines, and guanine, 8 guanine, and then 3-deoxyuridine 5 and 6 azuridine. These are basically used in the treatment of cancer. Then azathioprine is used as a suppressor in the immunological rejections during the transplantation. Then next is arabinosyl adenine is used in the treatment of neurological diseases and viral encephalitis. All these are nucleotides. Then arabinosyl cytosine is used in the cancer therapy as it interferes with the DNA replication. Then zidovudine or AZT is sugar modified synthetic nucleotide analog used in the treatment of AIDS. Thank you.